my name's Olivia Rowley and I do research on alarm pheromones and snapper. This research into snapper came about by a complete accident. Another researcher at our university was looking at vision in snapper, and they put a fish into a tank and caused a low level of stress, and then after taking the fish out, instead of changing the water, they put another fish in, and that nothing had happened to the fish, but it stressed the second fish out. So the question was raised of what's in the water that's causing this fish to be stressed. So my role, my research, was to investigate if this so-called alarm response actually did exist, and if it did exist, whether we could classify it behaviourally and physiologically and see the effects that it had on the fish. So to figure out the answer to my research question, we chose to use 3 different types of methodology. The first was looking at the behaviour of the animal, so seeing if when we injected the pheromone into the water, whether we could behaviourally characterise the difference in the fish, see what it was doing. The second method we used was looking at the fish's physiology, so we put little heart rate monitors on the fish and noticed that when the alarm pheromone was injected into the water, their heart rate peaked. And then the third method we used was super cool, in which we actually used fish tracking. So lighting a tank from the bottom, using the shadow, we could track the distance that the fish travelled, how fast it travelled, and just looking at whether that changed under the conditions of the pheromone. So what we found from the research was that behaviourally, in the presence of the pheromone, there was a difference in the fish. But what was super cool is that we found that not only was there your alarm response, so the fish increasing its swimming speed and where it goes and when, but also a secondary response that caused the fish to get these bars on its body and sink to the bottom and stay deadly still, which was super cool. We also noticed that not only was there a behavioural response, there was also a physiological response. So the heart rate in the fish peaked, meaning that it was in its flight response and it was stressing out. And also with the fish tracking stuff, we got to see things like pheromone avoidance, so the fish would avoid the area where the pheromone was in for up to 30 minutes afterwards, and also that the travel distance of the fish was increased, and its things like darting rate and surfacing rate were higher too. So what this means in terms of the overall biology of the snapper is that somehow, indirectly from a stress situation, one fish can let off a pheromone or an alarm cue that alerts all of the fish around this. So what this means is that it's probably an anti-predatory behaviour for the fish, and so when one fish is stressed or put in danger, it signals to the other fish that there's danger around. And so what this will cause the fish to do will be either A, get out of the way of danger, so freak out, which is the first response we saw, or it's time to hide. So bar up, sink to the bottom and stay still. So what was pretty novel with what we found was that in other studied fish, there has to be a laceration to the skin to let the chemical out. However, with the snapper, there was no damage to the skin. This was just caused by the snapper being inherently stressed. And so we don't know exactly where this pheromone comes from, whether it's something like excretion or whether they're voluntarily letting something out of the skin, but it's completely different and completely new, which makes it really awesome. So big picture wise, this research is super important because snapper is a really big fishery, especially in New Zealand where I did this research. However, history shows that the stocks are declining, and so the idea of growing these things in an aquaculture system isn't that far-fetched. However, in an aquaculture system, having all of these animals in close proximity to each other, one stressed fish can mean multiple stressed fish. And as you cannot be stressed and also grow and reproduce efficiently, being stressed out will affect things like how big you grow, the quality of your fillet, and also how fast you can reproduce and potentially up the population for the aquaculture system. What I hope to see this research used for is best handling practices for fish in closed water systems. So making sure that the fish have the best quality of care and also that they're growing well and reproducing well for the departments that are using them. My favourite thing about snapper is that they are a common fish in New Zealand. However, seen in the light of day, they have these beautiful luminescent spots along their back, and so they're just a really beautiful fish. What people would be interested to know about snapper is that whilst I looked at a species, particularly in New Zealand, that they are distributed throughout the whole entire world, so they're very common everywhere. What I love about being a scientist is the ability to look at the natural curiosities and quirks in the world and having the knowledge and the power to go and research them and understand why they occur.